in and thanks for joining me as we learn about the structure and function of membranes. In this lesson, you will learn the following topics. Keep in mind that you should feel free to pause and rewind any section of this lesson so that you can fully grasp the information presented. Membranes are primarily made of molecules known as fatty acids. Here's a diagram of two fatty acids. A fatty acid is a long chain of carbon atoms connected to each other, and each carbon atom is then connected to hydrogen atoms. In this case, the dark gray balls are the carbon atoms, and the light gray balls are the hydrogen atoms. Fatty acids have a special property in that one end of it is negatively charged. This end of it, because it's negatively charged, is known as a polar end. The polar end attracts water because water has similar magnetic properties. However, the long zigzaggy end, right, is called the non-polar end. And non-polar means that it's oily. So oil does not mix with water. So the oily end of the fatty acid chain repels water. So in this way, a fatty acid has one end that attracts water, the polar end, and one end that repels water, that is the non-polar end. Fatty acids can have a special feature in them, in that some of the carbons next to each other along the chain can be bonded together with a double bond. Okay, now a double bond does something very special to the fatty acid molecule, which we will talk about next. There is a nomenclature to describe whether a fatty acid chain is bent or straight due to a double bond somewhere along the carbon zigzag. If you draw a dotted line down the middle of the double bond, you will find that the hydrogens, in this case, hydrogen atoms, are either on the same side of the dotted line or on the opposite sides of the dotted line. Conversely, the same goes for the zigzag carbon chain part. Right? The zigzags are either both on the same side of the dotted line or they're on opposite sides of the dotted line. When the zigzags are on the same side of the dotted line, we call this the cis configuration, and cis means bent, okay? But when the hydrogen atoms or the zigzags are on opposite sides of the dotted line, we say that this is the trans configuration, trans indicating that the molecule is straight. Key terminology to know is that when we say a fatty acid is saturated, it means that there are no double bonds in the fatty acid chain. Okay, and the double bonds refer to double bonds between the carbon atoms in the zigzag region, and not, okay, I repeat, not the double bond between the carbon and oxygen, right, at the polar end. The term unsaturated means that somewhere along the zigzag line there is a double bond, and this double bond can either cause the fatty acid to be in a bent cis configuration or a straight trans configuration. When a fatty acid is named or called polyunsaturated, poly means multiple, right? Polyunsaturated means there are more than one double bond within the zigzag region of the fatty acid chain. Why is this important? It's because saturated straight fatty acid chains can pack together more neatly, and so they are closer in proximity to each other. This allows them to have a tighter packing. However, when a fatty acid has a kink in it, a cis formation that causes a bend, what happens is that the bent fatty acid, when next to the straight fatty acids, cannot pack together as closely. The result is that saturated fatty acids can behave more like one solid unit because like many books stacked together side by side. These cis fatty acids are bent so they can't pack together as well, resulting in a more fluid nature to membranes that have a lot of bent or cis configured fatty acids. of forces determine the packing of fatty acids when these acids are dissolved in water. One is called van der Waals forces. That is the forces between the carbon and hydrogen atoms in these zigzag lines. They attract each other. The other force is called hydrophobicity, or the nature of fearing water. Hydro means water. Phobia means fear. Hydrophobicity or hydrophobic means the fear of water. Since these long zigzaggy yellow regions are oily, they don't mix with water. And so what happens is that when they repel water, right, they will the oily regions will cluster together so that water molecules between them are pushed out, and water molecules can only be around the outer edges. Though membranes are made primarily of fatty acids, the fatty acids don't exist just as fatty acids. They exist as phospholipids. What is a phospholipid? The word phospho means containing a phosphate group, which is this group right here, a phosphate atom connected to 4 oxygens, and one of the oxygens has a strong negative charge. The word lipids and phospholipids means fat or fatty acids. Thus, a phospholipid is a molecule that contains a phosphate group that's negatively charged connected to 2 or 3 or even 1 fatty acid chains. In this case, phospholipids in the membranes of cells are phosphate groups connected to 2 fatty acid chains. Now, these fatty acid chains can be either both straight or 1 straight and 1 bent, or even, say, 2 bent fatty acid chains attached to a phosphate group. The red region here is known as the hydrophilic head. Hydro means water, philic means to love, meaning this red region here is attracted to water and likes to hang out with water. The yellow region here, the tails, are the hydrophobic or water-fearing parts. Hydrophobic is oily, and so these tails repel water and will seek each other out when dissolved in a solution of water, known as an aqueous solution. The phosphate group is connected to these multiple fatty acid chains through a 3-carbon molecule known as glycerol. 1, 2, 3. Glycerol is just a molecule that allows for multiple fatty acid chains to be connected to 1 phosphate group. The structure of phospholipids, one end water-loving, the other end water-fearing, causes phospholipids, when you drop them in, say, a bucket of water, to form these layered structures. The pink balls on the top and bottom are the hydrophilic or water-loving parts of phospholipids. They love interacting with water, and they, in effect, shield the yellow tails, the oily region, the hydrophobic region, from the water that is above and below. In this way, phospholipids can form phospholipid bilayers that separate water on the top from water on the bottom. On the left here is a ball model of what a phospholipid looks like. On the top part of this diagram is the individual atoms drawn out, so you can understand which atoms belong where, and each little dashed line is whether the 2 atoms are connected by a single bond, 1 line, or a double bond, as in this case between this carbon and this oxygen, there's 2 bonds between them. So we say that these 2 atoms are double bonded to each other, but that these 2 atoms are single bonded to each other because there's only 1 line between them. The components of a lipid membrane, especially one that is in cells, it's important to learn about a structure known as a molecule, known as cholesterol. Cholesterol is a type of fat. It's a sterol, right? It's a type of fat. It's oily, but it's not the same as a fatty acid. That is the yellow squiggly chains in these phospholipids. Cholesterol is special in that it's an amphipathic molecule. The word amphi means both, pathic means 2 states. So amphipathic means that cholesterol has a end that is polar, that is, likes to interact with water, and an N that is nonpolar, that is oily. So the nature of cholesterol makes it similar to the nature of phospholipids. One end polar, liking water, the other end oily, not liking water. In this way, cholesterol, right, is able to sneak itself into the phospholipid bilayer. Here is diagrammed as these green molecules, right, inserted into the phospholipid bilayer. And it can do that because it has one end that is polar, hangs out with the negative charge of the phospholipid, the phosphate group, while the long, it has a carbon chain, right, that is oily and can be inserted along next to the yellow fatty acid chains of the phospholipid. Cholesterol turns out to be very important in the function of membrane because cholesterol is able to prevent these fatty acid chains from sticking together too long. As in, if they stick together too long, if they stay next to each other too long, they might become a crystal solid, which would be bad for the cell, right? And so cholesterol, by floating around inside the phospholipid bilayer, is constantly bumping into these fatty acid chains, preventing them from sticking together too long that they become a solid crystal. In this way, cells can increase or decrease how fluid their membrane is by changing the concentration of cholesterol, how many of these green molecules are floating inside its membrane. For example, if the temperature gets really cold, things tend to freeze, right? In this case, bacteria can increase, in cold temperatures, bacteria can increase the amount of cholesterol, and this modification causes the membrane to be more oily and more resistant to freezing. Thus, even though the bacteria is living in Arctic temperatures, its membrane still remains fluid. When dissolved in water, phospholipids can form different types of shapes, given the nature of their two ends. The one end is polar, liking water. The other end is nonpolar, oily, and is afraid of water. The smallest structure that phospholipids form when dissolved in water is called the micelle. That is, it's a small sphere of phospholipids in which the polar heads are on the outside, shielding the oily tails on the inside from the water that surrounds it. When you have more phospholipids in the solution, the phospholipids can form what is called the bilayer, similar to the membrane of cells. And when there are enough phospholipids in solution, the bilayer can bend and connect and fuse, creating what's called a liposome. That is a vesicle or small pouch consisting of a phospholipid bilayer, right, with a hollow, empty, aqueous cavity on the inside. Aqueous meaning dissolved in water. So in this case, water is on the outside of the liposome and also on the inside of the liposome. The membranes of cells are not simply made up of phospholipids, though they are made of phospholipid bilayers. The membranes of cells contain many structures that are in it or stuck to it. These structures include proteins, here diagrammed in red color, sterols, which are a type of fat, okay, that's oily and can hang out with the oily tails of the phospholipids, right, but are themselves a different class of type of fat. In addition, there are sugar molecules, in this case, these light hexagons, right, these branching chains, these are sugar molecules linked together. These sugar molecules can be attached to a phospholipid, thus creating a glycolipid, glyco meaning sugar, lipid meaning fat. These carbohydrate chains, sugar chains, can also be bound to the proteins that are sticking inside of bilayers. In this case, you have a glycoprotein. Glyco, again, meaning sugar, protein meaning protein. The fluid mosaic model of the cell membrane, right, is meant to state that the membrane is not purely phospholipid, but consists of multiple different types of molecules. The word mosaic means a mixture, right? Means multiple pieces coming together to form a whole. Very similar to the individual pieces of glass that make up an image in a stained glass window. The word fluid suggests that the membrane is not a crystal, as in water frozen in the form of ice, but that the membrane is fluid, as in the parts can flow, just like water flows when it's at room temperature and is melted. So in this way, the proteins that are in the phospholipid bilayer can float around and move around the cell, which allows the cell to do many things, such as gathering a bunch of these red proteins next to each other, right, or excluding them from certain regions. A very important property of the phospholipid bilayer is that it is semi-permeable. So what is semi-permeability? The word semi means partially or half. Permeable means things can pass through it. So semi-permeable means only some things can pass through it. Phospholipid bilayers are oily in the middle, and so the only thing that can pass completely through them are things that are also oily, and otherwise known as nonpolar. Molecules that are polar, that is, that are electrically charged, whether negatively charged or positively charged, or molecules that have a water-like nature to them, meaning they like to hang out with water. Polar molecules cannot pass through the phospholipid bilayer because their polar nature will never allow them to sneak their way through the oily yellow tails of the phospholipid bilayer. Thus, this property of semi-permeability allows cells to be very selective of what goes in and what goes out, allowing cells to exist as we know them. Before we talk about the different types of transport across membranes, it's first important to talk about two concepts. One is diffusion, the second one is osmosis. So what is diffusion? Diffusion is the random and directional flow of molecules down their concentration gradient. A classical example of diffusion is here in the bottom left. You have a beaker of solution that contains red solute and blue solute, and say over time, say you put in the red solute, a drop of it at the bottom of the beaker. Over time, what happens is the red molecules will begin to move and spread throughout the beaker until the red and blue are equally distributed inside of that beaker. So that's a classic case of diffusion. A concentration of molecules going from a place of high concentration spreading out to a place where the concentration is now even in the whole system. Now, cells are different than artificial systems like this beaker because cells have this thing called phospholipid bilayers or membranes, which we've been talking about in this whole lesson. What membranes do is they add a level of complexity to this process of diffusion. So membranes allow cells to control what and how fast things move from one side of the membrane to another. And as we'll see as we go on in this lesson, this allows the cells to do incredible things. Why is diffusion random and directional? It's because in both of these systems, the artificial beaker and say this membrane separated system, even though the molecules say have spread out evenly on both sides, the molecules are constantly going back and forth. It's just that they go back and forth at the same rate, such that at any given time, it seems like, or there are the same amount of blue dots on top as blue dots on the bottom. Similarly, in this beaker system on the left, even though in this third beaker, the red and blue dots are evenly spread across the whole system, they're constantly moving around. They don't just stay in one place. Thus, diffusion is the random and directional flow of molecules. Right? Though molecules might have reached the state where they want to be equally spread out, whether in the whole beaker or just on either side, spread out evenly on either side of a membrane, they're constantly flowing back and forth in a random pattern. Osmosis is a special case of diffusion, and it exists because cells have membranes that are semi-permeable, meaning only certain things can pass through it. The semi-permeable membrane of cells results in two factors that affect diffusion of molecules, whether water or salt or sugars, back and forth between or across the membrane. Osmosis is the movement of water. Osmosis is both the directional diffusion of free water molecules from a place of high concentration to low concentration, that is down the gradient. Osmosis at the same time is also the directional keeping of water molecules on the side that has more solutes, okay, or molecules dissolved inside water. The classical case of osmosis is a beaker that contains a semi-permeable membrane separating two bodies of water. On the left here, this left body of water contains many sugar molecules, more than the body of water on the right side. So osmosis, what it does is that it will bring more water. It will bring water from the right side to the left side, as diagrammed here. And so the result is what? There's the water level on the left ends up higher than the water level on the right. In this way, there's almost a force, right? Or there is a force, a pressure that keeps the water level on one side higher and one side lower. In this case, we call it osmotic pressure, the pressure of osmosis. How is it possible that water will stay in this uneven, right, state, though it can freely pass back and forth between this membrane? Well, the reason is because osmosis has two forces at play. The first force is water molecules, okay, will want to go from a side that has a lot of it to a side that has less of it, that is from right to left. Secondly, water molecules, they can dissolve salt and sugar because what they do is they surround the positively charged molecules of the sugar or salt, separating these two molecules from each other, right? As long as there's water molecules surrounding this negatively charged particle and water molecules surrounding this positively charged molecule, these negative and positive charged molecules can't interact with each other. They can't form a crystal, and thus the sugar dissolves in water and disappears. What happens is that the side that has more sugar molecules has more water molecules that are busy separating these charged particles. And as long as they're busy separating charged particles, they're less free to flow back and forth. Thus, osmosis causes water to move to the side that has more salt or sugar, and that these water molecules stay on that side more often than they do going back to the other side. Thus, osmosis can cause water to stay on one side more often than the other, though water can freely pass between the membrane. When learning about osmosis and cells, it's important to know three key terms. They have to do with what's called tonicity. There are three states that cells can exist in terms of how concentrated the outside solution is. Isotonic means there's no difference. The concentration of liquid outside the cell is similar to the liquid inside of the cell, which is the definition of iso, meaning the same. The hypertonic state means that there is more solute molecules, salt or sugar, outside of the cell, and thus water will want to flow because of osmosis from inside the cell outside of the cell, which causes the cell to shrivel. The opposite case of hypertonic solution is the hypotonic solution. Hypotonic environments have more salts on the inside of the cell than it is on the outside. So the concentration of salt on the outside of the cell is lower than on the inside. So what happens is water wants to go inside of the cell to balance out the saltiness. And the result is that the cell will swell even to the point of bursting. Tonicity is important in understanding the reason why when you're in the hospital and your body's low on fluids, the doctors will inject saline solution into your bloodstream and not pure water. Injecting pure water into your bloodstream will kill you. To talking about the movement of molecules across a membrane, there are four types that we need to discuss. First is simple diffusion. Simple diffusion is also known as passive transport. As the word passive suggests, no ATP energy molecules are needed in order for passive transport or simple diffusion to happen. The second type of transport across a membrane is called facilitated diffusion. Facilitated suggests that the molecule is getting some form of help across the membrane, and that help comes usually from It's a protein channel that has a hole connecting the outside of the membrane to the inside of the membrane. Facilitated diffusion, like simple diffusion, does not require the ATP energy molecules. Active transport requires energy molecules called ATP, or adenosine triphosphate. The fourth type of transport is called gated transport. Gated transport means that there's a door on this protein channel that can open or close, preventing or allowing molecules to flow across the membrane through the channel. Gated transport can be facilitated diffusion, or it could be active transport, meaning you need to burn an ATP molecule in order to open that gate. There are two general types of gated transport channels. First is voltage gated, that is, you need an electrical current in order for the gate to open. Another is ligand gated channels. Ligand means a molecule has to bind to the gate, causing the door to open. Here's a diagram of the different types of transport. First is simple diffusion, that is, a molecule is oily enough to flow through the membrane by itself. Second is facilitated diffusion, that is, it is a one-way travel of molecules through a protein channel across the membrane. No energy molecules, no ATP is needed. Third is active transport, that is, this protein channel needs to burn an ATP energy molecule in order to move a molecule. Active transport is special in that it can move a molecule against its concentration gradient, meaning if there's a lot of molecules inside the membrane, diffusion won't let that molecule in by itself. However, the active transport, by burning an ATP molecule, is able to move a molecule into a region that has a lot of those molecules already. There are three classes of protein channels that are important to know in terms of moving molecules from one side to another of a membrane. Uniport is the movement of only one molecule across the membrane. Co-transport means two molecules are moved at once. If both molecules move from the same side to the other side together, this is called symport. If one molecule moves in while the other molecule moves out at the same time, this is called antiport. Cells are able to transport molecules in bulk, that is, in pouches, as opposed to individual molecules flowing across a membrane or through a membrane channel. The endomembrane system of cell are the membranes, or is the membrane, connecting the nucleus, the endoplasmic reticulum, the Golgi apparatus, and the plasma membrane, or the cell membrane. Cells can package large amounts of material inside a vesicle that is passed on from one membrane component to another, eventually delivering this packet of materials outside of the cell or to different parts of the inside of the cell. Because membranes can fuse with other membranes to form larger membranes. In this way, membranes allow cells to transport, export, and import large volumes of material through the outside of the cell or through the plasma membrane. And viruses can exploit, or they do exploit, this property of fusing membranes in cells in order to get inside of a cell. The same goes for the sperm when it interacts with the egg. It's four key terms to know in terms of vesicular transport: endocytosis, exocytosis, pinocytosis, phagocytosis. The classic example of how cells communicate with each other: two neurons talk to each other. If we zoom in on this synapse right here, what we see is that one neuron communicates to the other neuron by releasing chemicals that are contained in pouches inside the cell, outside of the cell. Once outside, in the space between the cells, the molecules float around until they bind to a channel on the other cell. This is the end of our lesson on the structure and function of membranes. Membranes are vastly important in many things in biology. You now have the foundations to understand the importance of membranes. Till next time.